So the picture you see of the neuromuscular junction is a good one, but you're not seeing the whole picture. This cell is a motor neuron, which is a cell that has a nucleus, a regular cell body, and many different specialized processes. One process I want you to know now is this long extension, this little telephone cable. It's called the axon, and it has a terminal that terminates this little bulb end. It's called the axon terminal. And this is the extension of the cell, the part of the cell that interacts with the muscle fiber at its surface. The muscle fiber, as we studied before the break, is a sarcolemma. And the part where the axon terminal communicates with it kind of pushes in a little bit. So this is the muscle fiber. And the part where they meet is the neuromuscular junction. Pretty much right here, there's a little gap where the two cells interact. It's called the synaptic cleft, or the synapse. It's a physical gap. Cleft. Physical space between the axon terminal and the muscle fiber. So we call this area a synapse. A synapse is a connection between two things. So we have two cells, a presynaptic cell and a postsynaptic cell. So sometimes they refer to the motor neuron as the presynaptic cell. If the synapse is the relative point. So then what would the muscle fiber be? The postsynaptic cell. And where they meet is the neuromuscular junction. Sometimes they don't call it the neuromuscular junction. Sometimes they call it the motor end plate, the part where the sarcolemma is a little more folded, where it interacts with the axon terminal. So I'm going to focus on the neuromuscular junction. And this group of slides starts off with a case study. However, we won't be able to fully address it today. So on Friday, I'll come back and we'll read through this together and talk about it as part of the lecture. But for right now, I'm going to get through the first few slides, because what we talked about before the break connects with this in terms of you got to get to the calcium release. This is what the last thing I taught before the break. Okay. So how do you get that nerve impulse to fire to stimulate the muscle fiber? That's why I want to talk about the neuromuscular junction now. Okay. And then on Friday, I'll talk about how it's called cross-bridge cycling. The specifics on how the actin and the myosin, they go through a cycle to get the whole muscle to shorten. Right. So for now, let's talk about this right there. You have to excite the muscle fiber.
Would it be an ATP?
No. The excitation is a chemical, but I'm not. What are we talking about? Someone said ATP.
Yeah.
What do you mean?
Like the energy to cause action?
You're right. ATP is going to be important for this picture here.
Okay.
But we're going to talk about this picture here for now.
Okay.
Well, here's a nice picture from the textbook. And, well, they kind of show you the whole central nervous system on the girl here holding a device. And what they're trying to show you is, maybe the device isn't that heavy, but you have this muscle that has to generate some tension to carry this device in your hand. And so there's, it starts in the brain, and there's a nerve in the spinal cord, and it sends this telephone cable, the axon, and it terminates on the muscle fibers. And so again, we're just looking at this part here. So this isn't the entire cell, correct? This is just the axon terminal. The cell body is actually inside the spinal cord. Okay. So this is just the axon terminal part on the muscle in the periphery. All right. So it's important to identify the steps where one cell triggers another cell. So basically what we're going to see, you're going to trigger an action potential. I'll draw it as a spike because that's how they're viewed in the lab. You're going to trigger an action potential in this cell, and it's going to be a strong enough stimulus to get that action potential to travel down the axon, and it will arrive at the axon terminal. And somehow, this action potential needs to spread to the postsynaptic cell. And how it does that, there's a chemical connection between the presynaptic and the postsynaptic cell. That chemical is acetylcholine in this case. I'll use like little green balls for acetylcholine. Abbreviated ACH. So there's a chemical communication between the presynaptic postsynaptic cell. That chemical is ACH, and the details you must know. So the details of that shown here. Let me go through the steps. So number one, the action potential arrives. So arrives where? Well, what is this called again? Axon terminal. The nerve impulse. Action potential arrives, and I'm putting in parentheses, I'm putting to the axon terminal. So the other thing is, well, what does it do when it gets there? It stimulates the release of that chemical, acetylcholine. So there's a second messenger called calcium. When the action potential arrives, there's voltage-gated calcium channels that open. Calcium is basically the signal to get these vesicles filled with ACH to fuse to the surface there. So I'll just put action potential arrives, you increase calcium levels. That is the signal to get the ACH released, step 3. Vesicles with ACH will then fuse to cell membrane. You're essentially, by exocytosis, releasing ACH. Releasing ACH, acetylcholine, into the synaptic cleft. Neurophysiologists, they call it a quantal release. It kind of releases in spurts. This fusion of ACH spilling into the synapse, quote unquote. Quantal release. Step 4, acetylcholine, a neurotransmitter, diffuses across the synaptic cleft and binds to its receptor in the sarcolemma. Right, that's the cell membrane there. Step number 4, ACH, a neurotransmitter. There's a class of molecules called neurotransmitters. ACH is one of them. We'll learn a few others in this class. Dopamine, epinephrine, there are other ones. But anyways, this one's ACH, the one that triggers muscle stimulation. Diffuses across the cleft. I'll just say diffuses across the synapse. And binds its receptor in the sarcolemma. Binds its receptor. Well, simply enough, the name of the receptor for ACH, we'll just call it the ACH receptor. We're going to call it ACHR for short. So ACH binds, I think that's receptor OR, sorry. It binds its receptor ACHR in the sarcolemma. So the ACHR, we call it a ligand-gated channel. Continue on here. Continued. ACHR is a ligand-gated channel. That means, well, what's the idea of a gate? A gate, like if you have a gate in a fence, how do you get through the fence? Well, I would go through the gate, right? You open the gate. And when we use it in physiology, we want students to know what triggers the gate to open. Okay? This class of ligand-gated channels, something binds it to make it open. In this case, the ligand is ACH, right? So maybe no, the gate's closed. But if ACH comes along, it fuses across the synapse, you have enough of it, and it binds its receptor binding site there, maybe it makes it open. That's the idea. That opens the gate. That's how it's gated. Now that the gate is open, a molecule, let's say sodium, sodium ion. Normally, sodium can't get past the hydrophobic zone. But if you have a gate, a protein channel that's ligand gated that allows for sodium to pass, well, now sodium can get through. Okay. Only when ACH is present. Remember, the green balls are ACH. All right, so that's what step 4 is. It's showing it to you there. It also shows the passage of potassium, excuse me. But just focus on the sodium influx. ACH binding opens ion channels that allows simultaneous passage of sodium into the muscle fiber and potassium out of the muscle fiber. That's step 5. So continuing on with step 5. Step 5. ACH binds ACHR, right? So ACHR, those channels open and it allows for sodium influx. Yes, you have the efflux of potassium. Focus on the sodium influx. We call this a depolarization of the muscle fiber at the motor end plate. So sodium influx depolarizes the post-synaptic cell. That's the key word. Sodium influx depolarize. Now, what we talked about in the first unit was how you have the sodium potassium ATPase pump. I had you write about it, and well, some of you wrote about it better than others, but we have to know that now. What that pump accomplished is you made the inside of the cell negative with respect to the outside. So imagine on the inside of the cell, it's lined with negative charges. And then on the outside of the cell, it's lined with positive charges. And it's almost like they're attracted to each other across the cell membrane. But the cell membrane is like our capacitor, it's our resistance. Okay. Things can't flow. Now, imagine ACH binds and the channel is open. We call this state here, where it's like not in equilibrium, but it's like all negative inside and positive outside. We call that, it's polarized. The cell membrane is polarized. But if this channel opens and you allow sodium in, which carries a full positive charge, you've depolarized it, right? You've depolarized the membrane potential. The thing about it is, these positive charges, it gets inside and it looks around and it sees nothing but negative charge. And it wants to spread there because opposites attract. Right? So what's going to happen is, this is going to flip. The positive charge, it's going to move. That's called current, when ions move. Right? It's a current. And these positive charges will displace the negative charges, so it's like, if I could extend this. Normally in rest, it's all negative on the inside. It's positive on the outside. It'll move in a way that flips it, so it'll look like it's literally moving. It's attracted to the negative charge it sees. And it's not just going to move in one, it moves everywhere in three-dimensional space along the surface of the sarcolemma. So any questions on the depolarization part? Sodium gets in, that's really important. But this is how you've stimulated the postsynaptic cell, that sodium influx. Okay, that's the action potential spreading from this cell to this cell. All right, so step 6 is cleaning it up. This will happen as long as ACH is present in the synapse. And muscle will keep contracting. So to stop the muscle from contracting, you have to get rid of the ACH. And there is an enzyme, ACHE. It's an esterase that breaks down ACH. That's step 6. So ACHE, E stands for esterase. Just think of it as an enzyme that breaks down ACH. So if ACH is removed from the synapse, the muscle can now relax. ACH removed. From synapse. Muscle relaxes. I'll teach some pictures of the model of the synapse, because I wanted you to know that for the quiz on Friday. So I'll come back to these slides. Just make a note. I'll come back. I'll explain all this. I don't have time now. I'll talk about the case study some more. But I had some pictures of the synapse, of the neuromuscular junction, to make sure it's clear to you. So if I ask you on a quiz on Friday, well, turn the lights back on. I want to mention one cell type that helps the removal of the ACH. It's called a satellite cell. Satellite cells, they help, they kind of soak up the excess neurotransmitter in these types of synapses. It's one of the functions. Help remove excess neurotransmitter from synapse. The reason I mention this is because one of our models actually shows a satellite cell, and you're expected to be able to identify it. Lights off. Okay, this is our axon terminal. And then so this is the presynaptic cell, basically. You have synapse there. You have some mitochondria. So these little vesicles, that's our ACH. Okay, the little yellow balls symbolize the ACH. Another picture. On this model, 7 is pointing to the nucleus. Well, it's a Schwann cell. I should mention that one too. Okay, 7 and 6 are another cell type called Schwann Cell. Let me write that on the board. It's got satellite cell, also note Schwann Cell.
And this is at the tail end of the ACH?
No. No. Satellite cell is because it helps remove neurotransmitter, which is kind of what I was talking about for 6. But we'll say that Schwann cell is just not part of this. So let me erase it.
Okay.
Erase.
Okay, sorry.
That's fine, that's fine. Yeah, I don't want to create confusion. There's a guy named Schwann from the 1800s, great physiologist. He discovered these cells. They help myelinate the axon. We'll talk about that in another chapter, but for now, know that Schwann cells, because it's shown here, they're not labeled Schwann cells, myelinate axons. Satellite cells, Schwann cells. That picture is showing you the Schwann cell and basically the Schwann cell. The myelin is kind of like right there, but I don't know how to identify that. But this right there, I'm pointing to very specific things here. For number 3, is pointing to the postsynaptic membrane, the sarcolemma. So you can call it either, if you call it sarcolemma, I wouldn't mark it wrong, that's correct. If you call it the postsynaptic membrane, I would accept that. Number 4 is the physical space. What do we call that? Well, it's right here, actually. Synaptic gap. Number 5, presynaptic
So
membrane.
that was the gram before one was the synaptic before?
Okay, it's right here. So number 4 is the synaptic gap. Number 5 is the presynaptic membrane. Okay, number 6, Schwann cell. Number 7 is the nucleus of the Schwann cell. This one I don't have a number for it, but what do you think it's pointing to?
ACH.
ACH. Yeah, ACH. Here's a picture of the model. What if I point to the blue thing? This tubule system. What do we call it? T-tubule. I could point to the blue stuff. What if I point to that hole? Same thing. T-tubule. Okay. What if I pointed to that? Terminal cisternae. Okay, remember that? What is that? Sarcoplasmic reticulum. Moving on. Axon terminal right there. Let's see. Oh, satellite cell. Okay. Satellite cell now model. That's why I like that. All right, so on your half sheet, we left off on 10, right? Yeah. Go to 11, 12, 13. On your half sheet. Go to 13. Number 11. Identify. That's 11. Little white balls. Number 12. Identify the blue thing. Blue structure. Number 12. Blue structure. Let's see here. Okay, last one. Number 13. Identify. Number 13. Identify okay. So for number 11, I pointed to that, right? What did you put? You should put ACH or ACH vesicle. Okay, same thing. Both are acceptable. For number 12, what would you identify this as? T-tubule. And then for the last one, which for this squiggly thing? Yeah, you should have put postsynaptic membrane or sarcolemma would have been acceptable. Okay. Let's shift gears in the lab. I got the models out, and what I did, I put like a key for the models. And people think that the key has some, are the answers for this. It's not. It's got nothing to do with it. Nothing at all whatsoever. But what I do want you to fill out these blanks. I will grade those. So I'm going to need 2 things from you. I need this filled out for the bone. And you can just rip off 1 page and just turn in the fill in the blank page for the muscle. So just turn it in and turn it in, and you're done for the day. Okay? Make 2 piles for me, whatever you're done, just right here. 1 for the muscle, 1 for the bone.
3 piles for the half sheet?
Oh yeah, yeah, we have a 3rd pile for the half sheet. I always forget that one. Even by the course, it's like